Welcome you all to the next class on inorganic chemistry of life, principles and perspectives. In the previous class I have talked to you about a few aspects of copper proteins, and copper proteins are categorized as you can see, the type 1 and type 2 kind of things, and type 3 and type 4, where you have type 1 plus T1 plus T2 plus T3, all of these are present. Then you have copper A, and then you have copper B, and then you have copper Z type. So, we have looked at all of these in the previous class, and then we have come to the copper 1 story, and as you can see that the copper 1 case. So, copper 1 centers, there are a few proteins structures are shown over here, and these are referred as a blue copper protein. And the blue copper protein, the copper in the blue copper protein, what you have is the blue color. The blue color is coming from the blue color is coming from the cysteine sulfur minus to the copper 2 plus charge transfer. So, this is a charge transfer transition. So, wherever you have a copper 1 site, you will find, or type 1 site, you will find this kind of a blue copper color. So, type 1 has nothing to do with the copper 1 state, copper 2 state, etc. So, therefore, this type 1 and copper 1 should not be confused. Now, as you can see from this slide that you have a few different copper site structures, and you see some more proteins over here. These are all type 1 containing ones. You can see in this protein you have the copper center somewhere in the towards the top, and mostly these proteins are more like a cylindrical in shape, all of these. The numbers that you see here 680, 375 to 85, etc., these are nothing but the redox potential of the copper that is present in these enzymes. That means rusticinin has got 680 millivolts, plastocinin has got 375 millivolts, azurin has 285 millivolts, stellocinin has got 185 millivolts, amicinin has got 260 millivolts, and pseudoazurin has got 255 millivolts. And you can see in the protein, this is all the protein moiety close to that, and this is the center where you have a copper and the binding moieties of this. So, on this slide you would see just the structure centers of the copper center only. This is again copper coordination sphere, all of these are you are seeing the copper coordination spheres. Now, here what exactly is bound? So, one does not need to remember these numbers like histidine 37, cysteine 84. All that one needs to know is there are two histidines, one cysteine, one methionine in plastocinin. And if you go to azurin, there is one glycyl CO 2 histidines, one cysteine, one methionine. So, this is the one where you have a 5 coordination, these are the ones where you have a 4 coordination. So, I have already told you in the previous class there are 4 coordinated ones, 5 coordinated ones, tetrahedral and the trigonal bipyramidal, but none of these structures, none of these geometries are or any close to the ideal kind of a structure. They are very highly distorted. I will just explain you once again the distortion, how it is helping. So, before going to the next point, let us look at these different values. So, the lowest being 185, the highest being 680 millivolts. So, a range of almost 400 millivolts difference or 500 millivolts difference. This total difference is coming not only just from the coordination sphere, but kind of a protein structure influencing all this. So, is that okay? Have you noticed the point number 1 is the redox potentials vary dramatically among the type 1 blue copper proteins, going from something like stellocinin 185 to something like rusticinin, which is 680 millivolts. Okay, let us look at a little bit more how such a distorted geometry will help. You see the kind of a distortion in plasticinin here. So, plasticinin here, if you see, this methionine bond is 2.85 angstrom, in other words 285 picometers, which is very far away from the normal one. The normal one in thiolate is about 2.10. So, during the redox activity, this is from a oxidized protein. Now, when the copper 2 plus receives an electron and reduces to copper 1 plus, then you get a reduced protein. So, therefore, electron transfer occurs between copper 2 plus versus copper 1 plus. Then when you have a copper 1 plus, what will happen? This particular methionine sulfur comes closer to this, and by at least about 0.4 angstroms, and one of these histidines go little farther away. So, that means distortion continues to be in a different way in the reduced form as compared to the oxidized form. So, what is that? Why is that kind of thing? So, that is because, as you know, that the copper 2 plus prefers more towards the square planar, and copper 1 plus prefers more towards the tetrahedral, and the original structure is neither the tetrahedral nor the square planar, it is somewhere in between. So, therefore, during the electron transfer, such a transition can take place. What transition? The geometry transition takes place very rapidly, therefore, you get a very high rate of electron transfer. In other words, during the electron transition, the reorganization of the protein is quite quick, and the reorganizational energy that is required is very minimal, so that it attains the new protein conformation compared to the old protein. That is, oxidized protein has one kind of a protein geometry here around the copper, and the reduced protein has a different kind of a geometry. So, switching over the geometry from one to the other is phenomenal in a sense that it requires very little reorganizational energy, as you know very well about the electron transition. During the electron transition, there is no change in the nuclear coordinates, and that is the kind of thing. So, you do not. So, quite quick reorganization. So, similarly, you can see another case, which is the azurin, is also quite well distorted case. So, all that I wanted to tell you is that the copper geometry in type 1 copper proteins is highly distorted, and during the electron transition, electron redox properties or electron transition, it goes from copper 2 to copper 1 very quickly, and the reorganization energy is not so high. So, therefore, it can easily reach the things. And just to show you an example, during the electron transfer, you can see that, and this is the kind of a protein that you have. This is the centers where you have a redox transfer, and you can see the other one too. So, it is just to give a feel that the redox transfer, electron transfer during the redox process between the two systems. This is a copper containing system, which is plastocyanine, and this is another system where the electron transfer is happening. So, here you have some kind of the receptors which are based on the porphyrin, and therefore, the electron transfer occurring. So, now we have seen that the type 1 has a very unusual geometry which is helping, it is a boon in disguise. So, why is that? Because during the copper 1 it should have one kind of a geometry favored, during copper 2 it should have another kind of a geometry favored, and these two geometries should interconvert at very rapid rate, and that is what is happening. Okay. So, having looked at this, now let us move to the next stage, which is the type 2 copper. So, this is type 2 copper, this has nothing to do with the copper 2. Now, type 2, one example I have shown here, it is the galactose oxidase. So, galactose oxidase has a type 2 copper, and type 2 copper center is also one single copper center. So, these are, the example is galactose oxidase. So, galactose oxidase does what? So, it converts the galactose to the corresponding aldehyde function. So, here what you have? Hydroxyl function, which is called the primary alcohol. What do you have here in product? Aldehyde. So, the alcohol to aldehyde. So, if you go back and look at what I talked to you earlier, and this is an oxidative reaction. How many electrons in this? I am sure you would have definitely identified that this is a 2 electron oxidation, but there is no oxygen number changing at all. CH2OH becoming CHO, and you get rid of 2 of the hydrogens in this. So, we will look into that. And in the process, there is no oxygen taken from outside at all, but in the process the oxygen which is used is converted to H2O2. So, this is quite different from the cytochromes. In the cytochromes it activates the O2, utilizes one of the oxygen and throws out the other oxygen as water. In dioxygenase both the oxygens are used, but here in the type 2 galactose oxidase the oxygen is not used for oxidation, but it is used to bring back the protein to the normal state. So, this is one thing. So, therefore, these are referred as oxidoreductase enzymes. So, it requires obviously a copper and it requires one another unusual thing. So, I will bring to you the unusual thing. You just look at the copper site coordination sphere and you have a histidine, another histidine, and this is a tyrosine and this is also tyrosine. But if you look at on this side there is something different than what you have. So, this part is coming from cysteine, this part is coming from the tyrosine, these two got coupled and this is the kind of a group which is not a original protein. It is a post translational modification that takes place after the protein binds to the copper center. So, when the copper binds to this region, this particular activation happens as a post translational activity and the whole thing is converted to a new cofactor, and this is the cofactor that you have in the galactose. And as I can show you in the next one, let us look at the protein structural features for a while. This is a huge protein having 639 amino acids and it has 3 domains. You can see the one the green domain, the blue domain and the red domain. So, this is you can call it as a domain 1, domain 2 and domain 3, and total 639 amino acids are broken into up to 155 amino acid residues are present in domain 1 and 156 to 552 is present in domain 2, which is a blue region here, and the 553 to 569 is present in a domain 3, which is red in color. And the domain 1 has got some sodium calcium binding sites, but most importantly it has something like the carbohydrate binding, because here what you are doing is you are taking the galactose and converting into the galactose oxidized form at the C6 position. So, therefore, the carbohydrate is important, the carbohydrate binding, carbohydrate recognition, that is there in the domain 1. And what is the domain 2? Domain 2 has got this type 2 copper center, that means catalytic center, that means reaction center, active center. So, all these is present in this particular the blue domain, and the red domain is domain 3 is a different thing. So, it forms some kind of a lid onto this one, because your active center is somewhere here and therefore, this forms like a lid and it provides one of the histidine binding, histidine 581, and that binds to the copper center. So, when the reaction goes on, initially the copper needs to be bound here, it will open up. When the copper is bound there, it will close it, and it can do both of these kind of things too. So, during the reaction it will close it. Once the reaction is finished, to remove the product it will open. So, the closing and open will be by the domain 3, and domain 1 is where the carbohydrate binding and domain 2 is the reaction center. So, as I said that the unusual cofactor that you have, and this is the cofactor, this one. So, one side cysteine, other side is tyrosine, okay, and this cofactor is the one which is stabilizes a radical when required in this. So, you can see fully oxidized enzyme have got copper 2 and the dot, copper 2 and dot, this is oxidized. Then when you add one electron, then this will be compensated, this radical, and then neutral, and therefore, you have a semi reduced, and then you add one more electron, that is the second electron, then that will become copper 1. So, copper 2 radical, copper 2 neutral, copper 1 neutral. So, fully oxidized, semi oxidized or semi reduced, either way is one and the same, and then fully oxidized. And this protein going from here to here is 2 electron the reduction. Then it should get back to the original, that is the 2 electron oxidation. Okay. So, this kind of a 2 electron oxidation means it gives away 2 electrons. When it gives away 2 electrons, what will happen to these 2 electrons? These 2 electrons are taken by the O2, and that O2 will become O22 minus, and with the presence of the 2 protons it will become H2O2. Okay. So, the external oxygen is not a part of this cycle, but the second half of the cycle to regenerate the galactose original form of it. Okay. If you look at the mechanism, you will understand and appreciate this aspect. See this is the resting state. Resting state, you have the binding of these ones, and then you have 1 tyrosine, 2 histidines, water, and this cofactor. And this cofactor in the resting part is not a radical, and this when it turns to the radical, that means 1 electron oxidation of this, 1 electron oxidation of this. So, that will be an electron cation here, and this is referred as the active form. So, once it comes to this, it is an active form. In an active form, the substrate will bind. What is substrate here? You are showing for easiness or CH2OH, because in the C6th position of the galactose you have a CH2OH. So, remaining thing we are showing like a R. So, therefore, do not be worried by this one. So, entire carbohydrate we are showing by R, and 6th position we are showing by CH2OH. So, this binds where? It binds in the position of the copper center by knocking out this water, knocking out. Knocking out means taking out the water, that is minus H2O, and this is the substrate bound center of this. Then the proton coupled electron transfer, and this proton whatever you have to the alcohol will go to this particular tyrosine, and tyrosine will get temporarily detached. Now, the proton coupled electron transfer will make this into the radical form. So, you see that the radical is formed at the substrate. This is the substrate. This is your alcohol, deprotonated alcohol, which is bound to the metal center, and that creates from here the thing is transferred over here, and the hydrogen which is present here is transferred over there. So, you see that this becomes OH, that means the radical is transferred from here to here. Okay. So, that is what is called the proton coupled electron transfer. And now, this particular thing which is the 1 electron radical, and if we transfer 1 more electron to that, to the copper will become the copper 1, and this the corresponding alcohol will become aldehyde, because already 1 electron is gone here, 1 electron is gone here. So, it will become copper 1 with the aldehyde out. The aldehyde out is shown as minus RCHO here, as you can see that. Okay. Now, the enzyme is fully in the reduced state, as you could see in the previous slide here, as you can see. So, it is the fully reduced enzyme, it has to get reoxidized. So, the reoxidation is taking place by this O2, external O2, and this O2 will bind to the copper, because copper is in copper 1 state. So, which is very hungry to take the oxygen, and takes up the oxygen and transfers an electron, then it becomes copper 2. Okay. And then again proton coupled electron transfer, and then radical goes back to the cofactor, and that will be basically into this, and that will give the H2O2. So, OOH and 1 more proton H2O2. So, that will give the active form. So, you can see that. So, actually speaking, your substrate is bound here and converted to product here, and the enzyme is in a different state, which is called the fully reduced state, and this reduced state is 2 electron reduced state, and this 2 electron reduced state is brought back to the normal state by O2, because O2 is capable of taking 2 electrons. The first electron will go to this one, the second electron will go to this one into out to make this particular radical and go back to this structure. So, can you understand that is a very easy kind of a thing. If you go through, if you follow me, it will be very easy. So, active form is radical form, and the water is displaced by the alcohol moiety or C6 position of the carbohydrate or galactose, and this is coupled with the proton transfer, proton coupled electron transfer from the cofactor, that will become copper 1, and the alcohol becomes the aldehyde and goes out, and this is oxidized by the O2, and the O2 is reduced by the thing to become the H2O2. Okay, so this is one example of the type 2. Let us look at another example of type 2. So, another example of type 2 is amine oxidase. So, amine oxidase is an enzyme. It is again a family of enzymes where it will oxidize the primary amine, it will also oxidize the diamines. So, this is also called primary amine oxidase, diamine oxidase. So, these are found in bacteria, fungi, plants, animals, all of these. These are somewhat smaller proteins, about 70 to 90 to 100 kilodalton, and these are homodimers. So, that means each one of the monomer has got the active center in these ones, and they have some 18 sandwich beta sheet kind of things, and this all you can read from this and find out. But the active site is buried and therefore needs a conformational change in order to access to the substrate or to remove the product, etc. So, what is the kind of a reaction? The reaction is RCH2NH2 and with the O2 going to RCHO plus ammonia plus H2O2. So, for this in the galactose oxidase you have seen one kind of unusual cofactor, and you will see a different kind of unusual cofactor which is called TPQ, topaquinone. TPQ is topaquinone. So, okay, you can see that this topaquinone is formed from the tyrosine residue, that you will see that. Okay. And this enzyme oxidizes the variety of biological amine containing neurotransmitters like histamine, xenobiotic amines, etc. It is also involved in a variety of metabolic pathways of amine moieties in glycine, serine, threonine, histidine, tyrosine, phenylalanine, tryptophan, beta-alanine, and also involved in alkaloid biosynthesis. So, the type 2 amine oxidase is a common one across a variety of things that you can see over here. So, the copper is coordinated in this to 3 histidines, to water molecules with a distorted trigonal bipyramidal, and has a dual function in making the topaquinone. So, now you can see huge size of the protein that you have. It is a dimer. In each monomer has got one catalytic center, and this is the catalytic center. You can see nearby amino acids and the catalytic center, this is your TPQ. The same thing is shown over here, nothing to worry about it. You can see the TPQ is labeled over there, and this is the TPQ which is very crucial for converting the amine finally in the reaction. So, how does it convert? Let us look at the next slide. In this slide you have the two things here, the two half type of reactions. In the galactose oxidase you have seen two half reactions. What are the half? First half is the galactose is oxidized, that is the first half. In the second half the reduced enzyme is reoxidized in the second half. So, now in this case also you have two halves, but a little differently in this. In this case the amine substrate is converted to aldehyde. This is the total reaction, aldehyde, ammonia and H2O2, but this goes by two halves. First half, the enzyme based carbonyl reacting with the substrate amine to form the Schiff base via this amine, which is very popular reaction, and oxidizing the amine to convert into aldehyde. So, this is obviously an oxidative reaction, but this is only half of the reaction, because at this stage the enzyme is reduced. So, the reduced enzyme is reoxidized by O2 and to get back to your normal state of this, and that will release the ammonia and the H2O2 also. So, at this stage the aldehyde is formed, but the ammonia is not released. For the release you need to reoxidize the enzyme. So, just like earlier case. So, you have a reduced oxidized form of the enzyme which converts the galactose alcohol into aldehyde, and then converts the reduced enzyme back to the normal form of the enzyme. In this case similarly, so you have Schiff base amine and aldehyde, and but then this is the ammonia is released out of this only in the second part of the enzyme. So, let us look at the first half of the enzyme, which you can call it as a reductive half. You can see that, and this is the cofactor TPQ part of it, and this interacts with your substrate, which is amine. As an example is shown NH2CH2R, and that will form a Schiff base, and this Schiff base is interconverted to this by losing the water and the amine moiety, and this releases the aldehyde. So, you have the aldehyde come, but still you have no ammonia seen yet. So, in the first half of the part, then you have this amine formation, but still bound to the TPQ. Aldehyde is only released, still ammonia is not released. In the second half, so this particular amine converted one is now undergoes further redox with the O2, and the O2 reacting with this enzyme and then converting the redox process between the TPQ and the O2 and converting into binding to the copper center, and then all these transformations from here it goes into this. So, that will release the H2O2 because you already made the O2 minus, and that will with the protonation will go to as H2O2, and still the enzyme is having one more stage, and this particular stage you have to get the ammonia out. So, to get the ammonia out you need the protonation for this, and that is by the water, and that will give the ammonium out. See. So, you have the total thing on this. Let us look at this one. So, first half and second half, and first half will give you the aldehyde and the amine bound one, second half will give the enzyme, the ammonia and H2O2. So, in that second half again you can see two parts here. In the first part the H2O2 is coming, the second part ammonia is coming. So, it is further, but anyhow this is the whole oxidative of, and that is the reductive half. So, putting together amine oxidase mechanism, you can see the enzyme, your substrate in the oxidized form of the quinone enzyme forming this Schiff base, converting into these ones, and losing the aldehyde, and then binding to O2 and then O2. So, the clarity of this is not shown here, but in the previous case that we have shown you how the O2 reaction goes here. So, in this this is not shown very well. So, take the previous one for this. So, you can look at the overall you get the H2O2 ammonia and of course, your aldehyde. So, what you get? You get in one stage aldehyde, in the second stage you get ammonia hydrogen peroxide plus the enzyme in the oxidized form. So, this is one which is in the process of type 2. Let us just begin with the type 3 before we conclude. Type 3 is again as an enzyme where you have the oxidizing the centers of like tyrosines. So, oxidized tyrosines will join together polymer, form a polymers, and these polymers will result in some molecules called melanin. You know, melanin is referred for color, black color kind of the skin. There are variety of melanins are there. This you can read, but I am not going to go into the thing. The one which is with the skin that you can see, and this particular thing is very important. Why? The black color of this is why is it very important? Because when the sun radiation impinges incidents on our skin, there will be some UV radiation, and this UV radiation should not damage the skin. So, that means this pigment should do something. So, it should not absorb, it should reflect or it should dissipate, and that is what it does, and that is why the people who do not have this pigment, they try to tan their skin, and that is what you see in western countries. The people tan their skin to get black pigment, and the black pigment will save you from the UV radiation. Otherwise, what will happen? And this radiation can cause cancer to the body, and therefore, the black color is boon in disguise. So, therefore, our skins in the Asia and particularly in India, our skin is very well suited less damage from the sunlight as compared to the western kind of thing. So, how does this melanin happen is basically tyrosine with a type 3 copper center. Type 3 copper center is oxidative. You see that, yeah. Dopamine and converted to this particular thing with this enzyme called tyrosinase. So, the enzyme is referred as a tyrosinase and it has a type 3 copper center. So, this will oxidize this and further slowly it will convert into these two. And so, this one the dopachrome, this is referred as a dopachrome and this is referred as dopaquinone. Why? There are two double bond O's here, that is called dopaquinone, and this is called dopachrome because one double bond O, one OH, and this one can form again in presence of the enzymes. It can lose the carbon dioxide and then form this diol kind of thing, and with one more enzyme again it can go back, and or even going through this one it can form. So, therefore, you can see variety of oxidative processes and this will make the eumelanin form of it. Okay. So, what we have seen in this particular class, I have brought to you a recapitulation of the type all different types of enzymes which I talked to you in the previous class, and then I talked to you a bit more on the copper 1 type 1 center, though I have talked to you in the previous class, I have again given you that. And then I talked to you about type 2, two examples, one is galactose oxidase, other is amine oxidase. Both of these are very important enzymes that I have explained to you. They have two cycles, one cycle is a half part, other cycle is the second half. In the galactose oxidase, the first part will oxidize the substrate and reduce the enzyme to, and then second part O2 will re-oxidize the enzyme. Similarly, in the amine oxidase, the first form you have a aldehyde is formed and the amine part that is attached to the cofactor TPQ will be further converted to H2O2 in presence of O2 and ammonia. And now just started with the type 3 where how the melanin is formed. So, I will explain the mechanism of type 3 in the next class. Thank you very much.